बिस्मिल्लाहिर्रहमानिर्रहीम। डियर स्टूडेंट्स, आज का जो हमारा लेसन है दैट इज़ अबाउट द बैक्टीरियल सेल ग्रोथ। वी हैव टेकन द एग्जांपल ऑफ बैसिलस subtilis. So growth have two means in bacterial life cycle. One mean is the irreversible increase in the size. If you look at this diagram here, these are the bacterial cells which have just formed through the binary fission, and they have undergone the growth. Now this is the fully grown bacterial cell. One of the fully grown bacterial cell then undergo further reproduction, which means binary fission or mitosis. Bacterial cells do not go under proper mitosis. They have another type of division which is named as binary fission, in which a cell simply divides into two. There are no prophase, metaphase, anaphase, or telophase because there is no nucleus. So there is no karyokinesis at all. So let's see what happens in binary fission or in the reproduction of the bacterial cell when there is plenty of nutrients, when conditions are favorable, and bacterial cell is having all the plentiful resources and better environmental condition, suitable environmental condition, then it will undergo binary fission. What will happen in binary fission? In the first stage, there will be growth. Complete size of the bacterial cell will be gained. All the enzymes, all the proteins needed for the next steps are prepared in this stage, which is named as the growth. Here it will be replication of the DNA. You see there is DNA molecule. This DNA molecule will replicate, and it will form two DNA molecules now. So these two DNA molecules in the next stage, they will migrate towards the poles of the mother cell. मदर सेल के पोल्स की जानब ये माइग्रेट करेंगे, एंड in the next stage what will happen? Cell membrane will invaginate from the center, and it will divide the cell into two equal daughter cells. So these daughter cells then will be segregated when we separated from one another. So here you see there is division of the mother cell into two daughter bacteria. दो एक bacterium जो है mother bacterium, that is divided into two bacteria. So you see there is a complete cycle of binary fission, in which you see there is very simple division. First of all, there is increase in the size of the bacterial cell. Then there is replication of the DNA. Then migration of the DNA molecule towards the opposite pole, and invagination of the cell membrane. And then the division of the cell. Cell divides into two. This is also named as log phase of the cell, log phase of the bacterial cell cycle. Here it only happens when there are sufficient or abundant supply of the nutrients. Nutrients, well, when are in plenty, then they can undergo binary fission, and this will continue. That's why bacterial colony increases in size in minutes. Then when there is insufficient supply of nutrients, then cell undergoes another process which is named as endospore formation or spore formation. In this case, what happens? When the bacterial cell DNA has been replicated, now the bacterial cell will not divide equally, rather there will be asymmetric or unequal cell division, and cell membrane will form a septum. This is the septum, and it will divide the cell into two unequal portions, and one bacterial DNA will migrate to this cell, and other is the mother cell. And in the next stage, there is a complete membrane formation around this DNA molecule, which is in very short cell, you may say. So it is named as pre-spore. It is also named as pre-spore, and the rest is the mother cell. Now the pre-spore will form another spore coat. It is the cell membrane, which was the mother cell membrane, and there is now coat pieces. These are, there is formation of the spore coat. So, and in between the spore coat and the cell membrane there is the cortex, and it is the mother cell. In the next stage, it is the completion of the spore coat, because spore will be a very resistant structure. Spore can withstand 100 degrees Celsius temperature in case of Bacillus subtilis, even in case of Escherichia coli or Clostridium. It can withstand 100 degrees Celsius boiling water for 20 minutes, and it can withstand 1 million rams of radiation. Radiation, gamma rays, are bombarded on the cell, and cells can be killed with the help of these UV light. But bacterial cell can withstand 1 million of rams. So you see in the next stage, when there is complete formation of the spore coat and there is complete formation of the cortex and cell membrane, then the mother cell disintegrates on only because there is less supply of the nutrients, so mother cell cannot survive. Only this cell now, which has become endospore or spore, that will survive. And spores can survive 20s of years in unfavorable conditions, and whenever there will be favorable conditions, these spores will germinate and they will produce new bacteria. In many cases, like in case of Clostridium and in case of dysentery and in case of tetany and some other bacteria, these bacteria remain inside the cell, and they remain there for many years. And whenever the conditions become favorable for the germination, especially when there is anaerobic conditions, why those people which do exercise, they get very less diseases, because they do not let these endospore to germinate. Because whenever somebody exercises, उसका जो टिश्यू होता है वो बेहतर तरीके से ऑक्सीजनेट हो जाता है, क्योंकि ब्लड सप्लाई बढ़ती है. और ये बैक्टीरियल एंडोस्पोर जो हैं, बैक्टीरियल एंडोस्पोर्स आर ऑलवेज प्रेजेंट इनसाइड आवर बॉडी एंड इनसाइड आवर टिश्यू, बट they will germinate only when there will be anaerobic condition, especially in the case of tuberculosis, in case of Clostridium, tetany, and some other bacteria. They will germinate only in anaerobic condition. So you must not let your tissue to be anaerobic. Let's exercise and give more supply to your cells, so that your tissue may not get anaerobic condition and endospore may not germinate. That's why I said that the people who are doing exercise, they will get less infection, because they do not let the endospore to germinate. And here the spore will remain for many years, and it can germinate after the arrival of the favorable condition. So this is the growth of the bacterial cell. Then we divide this growth of bacterial cell into 4 phases. When bacterial cells do not grow, they do not reproduce, then we say it is the lag phase. Then starts the log phase, and then here suppose it is the lag phase, and this is the log phase, when cells divide at exponential rate. Here it is the no growth phase. यहाँ पे मज़ीद जो है ना वो सेल्स ग्रो नहीं कर रहे. सपोज इट इज़ द कंडीशन इन विच द बैक्टीरियल सेल इज़ प्रेजेंट इन द फॉर्म ऑफ एंडोस्पोर और स्पोर, and when the conditions become favorable, when there is more supply of nutrients and conditions are favorable, pH, temperature, etc., and oxygen concentration is favorable for the bacterial cell, it will germinate and it will start binary fission, and cell will divide at exponential rate. It is the log phase. Then log phase, after the log phase, suppose you take the example of respiratory infection. फ़र्ज़ कीजिए हमारे रेस्पिरेटरी सिस्टम के अंदर एक एंडोस्पोर मौजूद है किसी बैक्टीरियल सेल का. यूं ही हमारे रेस्पिरेटरी सिस्टम में इस बैक्टीरियल सेल के लिए फेवरेबल कंडीशंस होंगी, न्यूट्रिएंट सप्लाई प्रॉपर होगी, तो इट विल स्टार्ट डिवाइडिंग एट एक्सपोनेंशियल रेट। यूं ही बैक्टीरियल सेल की ग्रोथ बढ़ेगी तो जाहिर है हमारा जो इम्यून सिस्टम है, हमारे जो वाइट ब्लड सेल्स हैं, फेगोसाइट्स हैं, दे विल आल्सो become activated, and our immune system will become activated, and immune system will produce more and more white blood cells. And although this is dividing at log phase, exponential rate, लेकिन अब क्या हुआ कि हमारे बॉडी के जो वाइट ब्लड सेल्स हैं वो भी बहुत ज़्यादा तादाद में बनना शुरू हो गए हैं और बैक्टीरियल सेल को वो फेगोसाइटोज कर रहे हैं, किल कर रहे हैं। तो एक वक्त आ जाता है कि जितने बैक्टीरियल सेल्स बन रहे हैं उतने ही बैक्टीरियल सेल जो हैं वो किल भी हो रहे हैं। सो इट इज़ स्टेशनरी फेज, जिसमें जितने जो डेथ रेट है, मॉर्टेलिटी रेट इज़ इक्वल टू द बर्थ रेट, तो इसको आप स्टेशनरी फेज कह सकते हैं। आफ्टर द स्टेशनरी फेज जाहिर है अब वाइट ब्लड सेल जो है, पूरा इम्यून सिस्टम जो है वो पूरी तरह से एक्टिव हो चुका है, एंड सपोज यू हैव टेकन सम एंटीबायोटिक एज वेल, जिससे बैक्टीरियल सेल की ग्रोथ जो है वो रुक गई है, बैक्टीरियल सेल की बाइनरी फिशन जो है वो रुक गई है, थोड़े बैक्टीरियल सेल पैदा हो रहे हैं और ज़्यादा बैक्टीरियल सेल जो है उनकी डेथ हो रही है। सो इट विल बी डिक्लाइन फेज, इसको फिर हम डिक्लाइन फेज कहेंगे। सो हियर आर 4 phases. First is the log phase, which is no growth phase. It is lag phase, no growth phase, and then second is the log phase, which is the rapid growth phase, जिसको आप कह सकते हैं एक्सपोनेंशियल रेट पे ग्रोथ होती है। And then there is stationary phase, in which the death rate and birth rate of the bacterial cell is equal. And then there is decline phase, when death rate is far more as compared to birth rate. So there is decline phase. These are the 4 phases of bacterial growth curve. It is also named as bacterial growth curve. You have already studied it. I just wanted to remind you people. So it is bacterial growth curve. That's all for today's lesson, my dears. Thank you very much. If you have questions, you are most welcome. Your question will be answered, inshallah. Allah Hafiz.